Good day, classmates. I'm Christine.
And I'm Reginald.
And we are here to demonstrate how to use the
Vis Spectrophotometer.
But first, what is UV-Vis? UV-Vis spectroscopy is a versatile, non-destructive analytical technique used to measure the absorption or transmission of light passing through a medium. It provides valuable information about a compound's electronic transitions. UV-Vis spectroscopy observes the absorbance of light energy or electromagnetic radiation. When light interacts with a sample, it excites electrons from the ground state to the first singlet excited state of the compound or material. And now we're going to discuss the basic parts of the UV-Vis. So we have here the Shimadzu UV1900i. So for its parts, here is the control panel. Ito yung control panel niya. So right now, standard mode siya. And touchscreen din po siya. And we have here the pen for the control panel. So, pipindutin lang siya dito to get the pen. For easier access of the control panel. And ibabalik rin lang siya dito. And we have here the lamp housing. So dito yung dalawang lamps which are the halogen and the deuterium. And here is the sample compartment. Can either use a multi-cell which is 6-cell or a dual cell. So ito yung dual cell niya. And ito yung dual cell niya. Right now kasi naka-install yung multi-cell or yung 6-cell. And so for this UV-Vis, yung right side niya, so dito nanggagaling yung light. While dito naman yung detector. So dito yung light and then dito yung detector. And we also have here the USB. So pwede siyang insertan ng USB para mag-store ng mga files. But remember lang, na-limited lang yung files na pwede niyang ma-store. So the control panel can be used alone kapag standalone lang na gagamitin yung UV-Vis.
So for this part, I'm going to teach you how to properly handle a cuvette. So this is a cuvette. A cuvette is used when using UV-Vis spectrophotometer. In a cuvette, we have a transparent part and the blurred part. We should always hold the cuvette in the blurred part. And we should avoid touching the transparent part because it might be damaged. So we use skim wipes to wipe the cuvette in order to make it clear and transparent before putting it to the multicell. So for this part, I will show you how to operate the UV-Vis spectrophotometer. First, we should plug in the wire. And we should turn on the automatic voltage regulator. And then after that, we should open the computer. And now we turn on the UV-Vis spectrophotometer. Now we wait for it to load. So after it load, we should tap the menu button on the lower left side and then proceed to PC control. So this is the multi-cell. Before using the multi-cell, we should remove the silica gel inside it. So in this multi-cell, we have 6 cells and 1 cell for the reference. Now we're going to put the cuvette in the UV-Vis. First, we should wash the cuvette 3 times by distilled water. Now I'm going to pour it some water. Then I will proceed to wipe it with skim wipes. and put it on the reference unit. We're going to do the same process on the second cuvette. Don't forget to always wipe it with the clean wipes. Now I'm gonna put it on the cell number 1. And now for this part, I'm going to use a 5 mm caffeine. Using a dropper, I'm gonna pour the caffeine into the cuvette. But first, I'm gonna wash the cuvette with the caffeine. And after that, I'm gonna pour the caffeine inside of the cuvette. And I will put it on the cell number 2. Now, to start the process of spectrum, we will first click on the connect button. And as we can see, I'm gonna click on the edit. Click on the accessories and make sure that it's set on the 6 series multicell. After that, I'm going to close after creating new parameter file. And I will name the file. The next thing I'm gonna do is click on the home button. Now, I'm gonna click on the one cell, and now it will start to load. So, for us to able to see the results, we need to click the active button, click on the peak, and click execute. As we can see, we receive the results over here. Now we're gonna try that process over again, but we should name all the cells. So this is the result for cell number 2. The cell number 2 contains the caffeine. So we're gonna put the results in here as a basis for our next mode, which is the quantitation mode. Click on the active, pick, and then execute. As we can see here, we get the result 273. Now, in order to save our file, we're just gonna click on the save. And it's already considered as saved. Now I'm gonna teach you how to use the mode quantitation. I'm gonna add another 2 cuvette containing caffeine of 10 ppm and 15 ppm. The droppers I've used on the 3 samples are different because they shouldn't mix. I'm gonna put it on the cell number 3. Same process on the 15 ppm caffeine. After that, we're going to click the quantitation button. On the quantitation button, we have here 2 graphs. So, what we're gonna do is click on the connect, and then and we click the edit in here. Click on the register wavelengths, and here we're gonna put the results that I've get on the earlier test, which is the spectrum. And click on the accessories. Always make sure that the accessories is set into 6 number of cell and 6 series of multicell. Close after creating new parameter file, and then name the file. Click on save, and then click on the edit file name, click on these 3 dots, and we're going to name it. After that, I'm gonna click on the... Wait lang, stop mo ulit. After that, I'm gonna delete one of these lines since our samples only include 4. Right click and delete line. Now we're gonna name all the samples. On the first part, we put water, so we're gonna name this sample as blank. On the standards line, I'm going to put their names. And after that, I'm going to put their concentration. So for the standard one, which is on the cell number 2, we have the 5 mm concentration. And here is the 10 ppm concentration and the 15 ppm caffeine concentration. After that, we are going to click on the instrument control and auto 0. After that, we're gonna click on the standard. And as we can see, the results over here and the graph will show. So for this part under here, this type is the unknown, and we're just gonna set on the cell number. For example, I'm going to click on the cell number 3. I'm going to click next. So we are now on the cell number 3. Assume that we don't know what type of the sample that we have there, what concentration of the sample that we have there. We are gonna click on the 1 cell and it will show the concentration. Now we are going to proceed to photometric mode. On the photometric mode, all the samples are unknown, unlike in the quantitation mode. We are going to delete the excess lines, and we're gonna start by clicking on the connect button. So this is the instrument control. I'm gonna first edit, and again, let's put the results that we have on the first part, which is the spectrum. Accessories. Make sure that it's always set on the 6 series multicell, and close after creating new parameter file. Now we name the file. And edit file name. Click on the 3 buttons here. And open. Now we're gonna click on the home. And as we can see, this is cell 1. I'm gonna click on the auto 0. After clicking the auto 0, we're gonna proceed by tapping the unknown sample. So as we can see, the results are over here and the sample graph are over here. So that is how to use all the 3 modes of the UV spectrophotometer.
And that's all for today.
I hope you learned something.
Thank you for watching.